Imagine, if you will, two young patients in a pediatric oncology ward.
Okay.
And on paper, their medical charts look utterly identical, you know? They've both just been diagnosed with the exact same type of aggressive blood cancer.
Right, T-cell acute lymphoblastic leukemia, or TAL.
TAL. And they are prescribed the exact same grueling regimen of standard chemotherapy. Fast forward a month, one patient is declared in remission, essentially cured.
Which is the goal, obviously.
Right. But the other patient, their cancer it shrugs off the drugs completely, and it leads to a devastating, often fatal relapse.
It is the absolute definition of a medical paradox. We have two identical diagnoses, but completely divergent realities.
Yeah.
And for decades, this has been the agonizing reality for doctors treating TAL. You look at the cells under a microscope, and they look the same.
Yeah.
But clearly, something invisible is determining who lives and who relapses.
Right. And the answer to why this happens, it isn't found in the amount of cancer the patient has. It's not about tumor volume.
No, not at all.
The secret, it turns out, is hiding in a sort of cellular time machine inside the bone marrow. So, okay, let's unpack this. Our mission today on this deep dive is to tear into a really groundbreaking November 2024 paper from Nature Cancer.
And the scale of this research is just staggering. They didn't just look at a few tumor samples.
No, they mapped out over 595,000 individual leukemia cells to solve this exact clinical mystery.
Which is, that's a massive data set.
Because treating relapsed TAL is incredibly difficult. The salvage options are, well, they're slim to none. Doctors desperately need a way to look at a patient on day zero, like at the moment of diagnosis, and know immediately if that standard chemotherapy is going to bounce right off the tumor.
Exactly. And to understand what is fundamentally broken in a leukemia cell, we first have to understand what the machinery looks like when it's working perfectly.
Makes sense.
So the researchers started by building an incredibly detailed multiomic reference map.
A healthy atlas.
Right. They took healthy pediatric bone marrow and thymus tissue, which are essentially the biological factories where our blood and immune cells are manufactured, and they mapped the entire developmental hierarchy of blood formation.
Now, mapping 595,000 individual cells sounds like, science fiction to me. How do you even look at the inner workings of a single microscopic cell?
Well, they use two primary techniques, scRNA-seq and scat-tac-seq.
Okay, I think I've got a handle on the first one, single-cell RNA sequencing, or scRNA-seq. Basically, if DNA is the master blueprint stored in the cell's nucleus, RNA is the active photocopy of the instructions the cell is using right now. So scRNA-seq lets us read a single cell's active to-do list, right?
That's a really great way to put it. It tells us which specific genes the cell is actively translating into proteins at that exact millisecond.
Wow.
But to get the full picture, they paired it with scat-tac-seq. This stands for single-cell assay for transposase-accessible chromatin.
Okay, hold on. That is a serious alphabet soup. Transposase-accessible chromatin. Break that down for me because I am totally lost.
Fair enough. Let's visualize the DNA inside a single cell. If you stretched it out, it would be about 2 meters long.
2 meters in a microscopic cell.
Yeah, exactly. So to fit inside that tiny nucleus, that DNA is tightly spooled around proteins called histones, almost like thread wrapped around a wooden spool.
Okay, I'm picturing that.
When DNA is spooled up tight, it's closed. The cell can't read it. Scat-tac-seq identifies the physical areas of the genome that have been unspooled, the areas that are opened up.
Oh, I see. So if the genome is a massive multi-story library, most of the books are locked in the basement.
Right.
Scat-tac-seq shows us which specific doors have been unlocked and left open, even if the cell isn't actively reading the book yet. It shows us what the cell is capable of doing.
Spot on. It reveals the cell's potential. So by combining the active to-do list with the unlocked potential, they built this healthy reference map.
I love visualizing this healthy map as like a massive public school system.
Oh, that's a good analogy.
Yeah, so think about it. Every blood cell starts out in preschool as a totally unformed stem cell. Over time, it grows up. It graduates to elementary school as a multipotent progenitor.
Then middle school, high school.
Until it finally graduates as a fully mature, functioning adult T cell. And what the researchers found when they took the blasts of the cancer cells from 30 different TAL patients and mapped them onto this healthy school system is that leukemia is fundamentally a disease of arrested development.
Yes. The leukemia blasts literally get stuck in a specific developmental grade. Yeah. And instead of growing up and graduating into useful immune cells, they just sit in that grade and endlessly divide.
making millions of identical immature copies of themselves.
Exactly. And the really wild part is that even within the exact same official clinical diagnosis, say a subtype called early T-cell precursor AL or ETP-OLAP,
Right.
the patient's cells were resting at completely different grades. Some patients had tumors entirely stuck in preschool, while others were stuck in middle school.
This is a major paradigm shift, isn't it? Because the traditional way we classify these tumors is by, staining them and looking at proteins on their surface under a microscope.
Yeah, flow cytometry.
Right. But this single-cell mapping proves that our standard diagnostic tools are missing the underlying developmental reality. Two tumors that look identical to a pathologist might be operating on completely different biological software.
Exactly. Which changes how we have to approach treating them.
So if you're tracking with us so far listening to this, let's do a quick recap to cement this idea. TAL cancer cells aren't just rogue cells. They are cells frozen in a specific stage of development.
Right.
And scientists use these incredibly precise molecular tools, checking the cell's active to-do list and its open library books, to pinpoint exactly which grade each patient's cancer was stuck in.
Yes.
Which really begs the question for anyone listening. If you're a doctor, how does a patient whose cancer is stuck in preschool react to treatment compared to a patient whose cancer made it all the way to high school?
That is the pivotal question.
Right.
And it's exactly where the researchers looked next. They focused on the patients who failed chemotherapy.
Right.
In pediatric oncology, this is evaluated by measuring minimal residual disease, or MRD, at day 29 of treatment. Day 29 marks the end of the very first intense phase of chemotherapy, which is called induction.
Induction is basically the shock and awe campaign, right?
Yeah, precisely.
The goal is to get the cancer down to 0. So if you hit day 29 and you still have cancer cells hanging around, meaning you have high MRD, the shock and awe didn't work. You're in an incredibly dangerous territory.
Exactly. So they looked at the single-cell maps of those specific treatment-refractory patients, and a glaring pattern emerged. The patients who failed therapy all harbored a high concentration of a specific subpopulation of cells called BMP-like cells.
BMP stands for bone marrow progenitor, so these are the preschoolers.
They are the absolute earliest stage. In healthy biology, these are equivalent to hematopoietic stem and progenitor cells, or lymphoid prime multipotent progenitors.
Okay, more heavy terminology. Let's translate that. A multipotent progenitor is basically an undeclared freshman. It hasn't even picked a major yet.
Right.
It hasn't committed to becoming a T cell or a B cell or a red blood cell. It is totally primitive.
Yes. And conversely, the patients who were cured by the chemotherapy had cells that were stuck much later in development. They had declared their major. The researchers called these T-specified cells.
The high schoolers.
The high schoolers, exactly.
So we've unmasked the villain here, the primitive BMP-like preschool cell. But why on earth does being stuck in preschool make a cell bulletproof to chemotherapy? I mean, a cell is a cell. Poison should kill it, right?
You would think so.
Yeah.
But it all comes down to a specific molecular mechanism involving a gene called NR3C1.
NR3C1.
Got it. The frontline weapons we use against pediatric leukemia are corticosteroids.
Yeah.
Primarily a highly potent steroid called prednisolone. But prednisolone doesn't just melt the cell from the outside. To work, it has to enter the cell and physically bind to a specific internal receptor known as the glucocorticoid receptor.
And that receptor is manufactured by the NR3C1 gene. Okay, I think I see where this is going.
Go ahead.
If you picture the cancer cell as a heavily fortified bank vault, prednisolone is the key. But if the cell isn't actively expressing the NR3C1 gene, it means it never bothered to build a keyhole in the vault door.
That's a perfect visual. The primitive BMP-like cells naturally have incredibly low expression of NR3C1. They haven't built the keyholes.
Wow.
So the prednisolone molecules are just floating through the bloodstream, bumping into these BMP-like cells and bouncing right off. There's nowhere for the drug to bind.
While the older T-specified high school cells do have the keyholes, the drug enters, binds to the receptor, and triggers a self-destruct sequence inside the cell, wiping them out.
Exactly. And the lab results confirming this are staggering. They tested this directly, and the BMP-like cells showed over an 80-fold increase in resistance to prednisolone compared to the mature T-specified cells.
Wait, 80-fold?
80-fold. Just because of what developmental grade the cell is stuck in. Furthermore, because these BMP-like cells are so primitive, they naturally express genes that allow stem cells to endlessly renew themselves.
It's the ultimate recipe for a relapse. I mean, put yourself in the doctor's shoes for a second. You hit the patient with standard steroids. The chemo successfully wipes out all the mature high school cancer cells, which makes up the bulk of the tumor.
Right. The scan looks clear.
Yeah, the patient's blood work suddenly looks fantastic. But lurking in the bone marrow is this tiny, hidden reservoir of preschool cells that completely ignored the drugs. And because they have that stem cell power of self-renewal, they slowly rebuild the entire tumor from scratch.
So the fundamental challenge shifts. If our standard trusted weapons are physically incapable of unlocking these primitive cells, how do we adapt?
Right.
To figure that out, we need to know why a cell gets stuck in preschool in the first place, while others manage to mature into the vulnerable high school stage.
And here is where the paper delivers a massive plot twist. It all comes down to genetics. Normally, when we talk about cancer, mutations are the bad guys.
Always.
They're the broken brakes or the jammed accelerators driving the disease. But in this specific scenario, certain mutations are actually doing the heavy lifting to save patients' lives.
It's a fascinating tug of war. The researchers took their single-cell data and intersected it with the cell's DNA. They found that the deadly, primitive BMP-like state is largely held in place by genetic fusions involving the HOXA gene cluster.
Okay, let's clarify a fusion gene for the audience. This is when two completely separate pieces of DNA break apart and accidentally fuse together incorrectly. Like the MLLT10 or KMT2A fusions mentioned in the paper, it creates a mutated sort of Frankenstein protein.
Correct. And these HOXA fusions act like a massive, heavy anchor. They lock the cell down in that primitive, undeclared multipotent state.
Okay, so the fusion is the anchor holding them in preschool.
Yes. But then they analyzed the mature, highly treatable T-specified cells. And they found those cells were driven by a different mutation, a mutation in a gene called NOTCH1.
NOTCH1.
Now, NOTCH1 is a critical component in healthy biology. It creates a receptor on the surface of the cell that acts as a master switch. When a healthy stem cell is floating around and suddenly receives a NOTCH1 signal, it is a biological mandate. The cell is instructed to abandon all other career paths and strictly mature into a T cell.
In TAL leukemia, that NOTCH1 gene is frequently mutated so that the switch is permanently glued in the on-end position. Now, instinct tells you a hyperactive mutated master switch. That must be making the cancer worse.
Naturally, yeah.
But the researchers used an incredible technique to look closer.
Yes, a technique called genotyping of transcriptomes, or GOT. It's not enough to just know if a mutation is present.
Yeah.
GOT allowed them to look at the exact dosage of the NOTCH1 mutation inside a single cell. Every gene in our body has two copies, or alleles, one from each parent. They found patients whose cancer cells had distinct NOTCH1 mutations on both alleles, a double dosage.
Wow. So if the HOXA fusion is the heavy anchor holding the cell in preschool, this double NOTCH1 mutation acts like an overpowered engine pulling it forward.
That's exactly it.
The NOTCH1 mutation is this incredibly strict biological guidance counselor. Even though the cell is cancerous and just wants to sit in preschool and multiply, the NOTCH1 mutation grabs it by the collar and says, no, you are going to high school. You are going to differentiate.
And by physically forcing that cancer cell to grow up, the NOTCH1 mutation forces the cell to change its gene expression. It inadvertently forces the cell to build the NR3C1 keyholes we talked about earlier.
Oh, wow.
It accidentally makes the cancer cell highly vulnerable to the standard steroid chemotherapy.
The mutation that is driving the cancer is simultaneously painting a giant target on its back.
The clinical data perfectly validates this too. In their bulk sequence cohort, they identified 18 patients who had 2 or more NOTCH1 mutations. Every single one of them, 100% of those 18 patients survived.
That is just incredible. So to pull this all together for everyone listening, the fate of the patient depends on a genetic wrestling match inside the cell. The HOXA fusions try to keep the cell in a dangerous primitive state that entirely resists chemo.
Right.
But the NOTCH1 mutations act as an engine, forcing the cells to mature and build the very keyholes that allow our drugs to get inside and destroy them. I mean, think about how that flips our traditional understanding of tumors upside down.
It really does.
We spend billions of dollars in oncology trying to fix or block mutations. But here we have a hyperactive cancer mutation that is actively doing the doctor's job for them. It completely redefines what a bad mutation is.
Exactly. But it still leaves us with a critical clinical problem. We know the villain is the primitive BMP-like cell, We know why it survives. It lacks the keyholes for steroids, and we know the genetic tug-of-war that creates it.
Right.
But how does this help the kid sitting in a hospital room right now? You can't run a multi-million-dollar 595,000 cell sequence on every new patient who walks through the door.
Right. They needed to turn this laboratory masterpiece into a cheap, rapid tool a doctor could actually use in the clinic. So they used machine learning to distill all that massive single-cell data down to its core essence. They found a specific signature of just 17 genes that perfectly identifies this dangerous primitive state.
Yes, they named it BMP-17.
BMP-17, got it.
It acts as a universal flag for treatment resistance. A hospital can run a standard routine bulk RNA test on a patient's bone marrow on day 0. They look for this 17-gene signature, and they immediately know if the patient is harboring a reservoir of those deadly preschool cells.
That's huge.
And importantly, this BMP-17 signature successfully predicted poor survival across all subtypes of TAL, proving it's a universal vulnerability.
Okay, so we essentially have a molecular wanted poster. We can spot the resistant cells on day 1, but, identifying a bulletproof cell doesn't do you much good if you don't have a weapon that can pierce the armor. How do we kill it?
To find a new weapon, the researchers created PDX models. That stands for patient-derived xenografts. In plain English, they took these specific human BMP-like leukemia cells and engrafted them into immune-deficient mice. This creates a living, breathing biological testing ground. Then, they ran a massive therapeutic screen, systematically throwing 40 different active leukemia drugs at these human cells growing inside the mice to see if anything worked.
And they found a massive vulnerability. These primitive blasts are incredibly sensitive to a class of targeted drugs called BCL2 inhibitors, specifically a drug called venetoclax. But before we explain why venetoclax works, we have to explain what BCL2 is.
BCL2 is fundamentally about cellular survival. Every single cell in your body has a built-in self-destruct mechanism called apoptosis.
Right, apoptosis.
If a cell gets too damaged or infected, the mitochondria, the powerhouses of the cell, release a signal that essentially tells the cell to gracefully dismantle itself. BCL2 is a protein that acts like a heavy safety cover placed over that self-destruct button. It tells the cell, do not die, no matter what.
And because healthy stem cells are incredibly precious, we need them to last our entire lives, they naturally produce huge amounts of BCL2. They have extra safety covers on their self-destruct buttons.
Precisely. And because these BMP-like leukemia cells are essentially just arrested stem cells, they inherit that exact same biological software. They are absolutely addicted to BCL2 to survive.
So venetoclax enters the chat. It's a targeted inhibitor. It swoops in, binds directly to the BCL2 protein, and physically neutralizes it. It literally rips the safety cover right off the self-destruct button.
Once that BCL2 cover is removed, the primitive cancer cell can no longer hide from its own internal damage. The mitochondrial threshold is instantly breached, and the cell undergoes rapid apoptosis. It self-destructs.
Wow.
The vault door might not have a keyhole for steroid treatments, but venetoclax doesn't need a key. It just detonates the hinges from the inside.
The results in the live mouse models were astounding, right? When they gave venetoclax to the mice harboring these high BMP leukemia cells, it didn't just slow the cancer down. It cleared the disease to undetectable levels in both the bone marrow and the spleen. It completely eradicated the root cause of the relapse.
This research paves the way for a completely revolutionized clinical algorithm. Imagine the near future. A child is diagnosed with TAL. Instead of blindly starting them on toxic steroids and just crossing our fingers for 29 days, we test their bone marrow on day 0 for the 17-gene BMP signature.
If they score low on the BMP signature, great. It means their cancer is stuck in high school, they have the keyholes, and you proceed with the standard, highly effective steroid therapy.
Exactly.
But if they score high, if they're harboring the preschool cells, you don't waste 29 days poisoning a child with chemo that you mathematically know won't work. You bypass the steroids entirely and hit them immediately with a targeted BCL2 inhibitor like venetoclax to rip the safety covers off those cells.
It is the absolute pinnacle of precision medicine. We are moving away from treating a cancer based on its microscopic appearance and moving toward treating the exact developmental and molecular vulnerability of its most dangerous cells.
Let's lock in this final piece of the puzzle. So scientists took 500,000 data points and built a 17-gene wanted poster so doctors can spot drug-resistant cells on day 1.
Right.
Then they figured out that because these primitive cells are addicted to a survival protein called BCL2, a drug called venetoclax acts as their specific molecular kryptonite, forcing them to self-destruct.
It's an incredible leap forward, but it also leaves us with a really profound concept to consider as we wrap up. We spent a lot of time discussing the NOTCH1 mutation and how its double dosage acts as an engine, physically forcing primitive cancer cells to mature and build the receptors that make them vulnerable to our drugs.
Right, it forced the cancer to grow up.
This raises a provocative question about the entire future of oncology. Traditionally, cancer treatment has always been a game of poison, finding a toxic chemical that hopefully kills the cancer cells just slightly faster than it kills the healthy tissue.
Yeah, that's how it's always been done.
But what if the future isn't about poisoning cells at all? What if the next great frontier is differentiation therapy?
Oh, wow. Meaning inventing synthetic drugs that act exactly like that NOTCH1 mutation.
Exactly. Drugs that don't try to kill the cell directly, but simply latch onto primitive, highly resistant cancer cells and forcefully trigger them to mature. If we can invent a drug that forces a leukemia cell to grow up and graduate high school, it naturally loses all of its stem cell armor.
That makes total sense.
Once the armor is gone, we can easily sweep the cancer away with the mild, standard medicines we've had for decades.
Using the cancer's own developmental programming against itself. I mean, we've gone from looking at a murky, confusing clinical picture where treatments seemingly failed at random to possessing a molecular map that literally points the way to a cure. It changes everything.
It really does.
Well, thank you for joining us on this deep dive. Take these ideas, chew on them, and remember that the shortcut to being truly well-informed is always questioning the standard narrative. Until next time.